Hello everyone, my name is Jimena Ibagon and I'm a visiting scholar working with Dr. Hans Stein at the University of Illinois. Our lab focuses on determining values of digestibility of energy and nutrients, and today I'm going to talk about how sunflower experllers have greater ileal digestibility of amino acids than sunflower meal when fed to growing pigs. So to begin with, I would like to discuss sunflower and the importance of conducting research using it. Over the past few years, sunflower oil is extracted from sunflower seed mainly for human food consumption. An important fact about the position of sunflower in the oil industry and feed industry is that in 2020, global sunflower seed production was to be around 54 million metric tons, making it to be the third most produced oilseed around the world after soybeans and rapeseed. Noticeably, sunflower seed has high content of oil. Therefore, oil production from sunflower has reached 19 million metric tons during the last 5 years. And sunflower meal, as another important byproduct of sunflower seeds, has been trending up 22 million metric tons in the global production. Countries including Ukraine, Russia, and the European Union are the largest producers of sunflower seeds, but we can include, based on the USDA data, countries as United States and Argentina that have a significant production as well. There are two important methods of oil extraction in sunflower meal, and one of them is the expeller pressing method that I'm going to further explain using this schematic representation. The process starts with the cleaning and storage of the seeds, followed by the dehulling process. The dehulling process involves partial or total removal of the hulls from the seeds, and this is a very important step because it affects the final quality of the meal because of the high fiber content on those hulls. After that, we have the crushing rolls, which is the first press of the sunflower seeds, and then the seeds are going through the final pressing, which sometimes involves high temperatures because of the friction. After that, we finally obtain the sunflower expeller. In some crushing plants, there are just one pressing step before obtaining the oil. And after the meal is separated from the oil is being filtered and centrifuged to be degummed. Sometimes these gums are added back to the meal in the final process, but this step may vary among processing plants. After that, the oil is ready for the final purpose. The second oil extraction method is the solvent extraction process, which is the continuation of the expeller pressing process, but to this process the solvent extractor is added. So, after the first crushing of the seed, and in some crushing plants after the second one crushing, the seeds are going through the solvent extractor. And this step is to obtain more oil from the meal. The meal is being desolventized to finally obtain the sunflower meal. Therefore, those are the difference in the processing and the way to obtain those co-products. And basically, the difference between sources could be the addition of the third step, which involves the solvent extraction. Talking about sunflower co-products, we can say that sunflower meal is one of the most important co-products of sunflower seeds. This is because it has many good characteristics, which makes it an ideal byproduct to feed pigs. First of all, sunflower meal has been used as an alternative and less expensive source of amino acids than soybean meal, which can be fed to swine and poultry. However, even if we know that sunflower meal has a high content of protein, the nutritive value and quality of the meal may vary due to the variation in climate conditions, soil, cultivation methods, place of growing, and difference in the oil extraction process. One of the most important steps which may affect the composition or quality of the final sunflower meal is the degree of the dehulling, or the addition of hulls back to the meal in the oil extraction process, which may increase fiber content in sunflower meal. Therefore, to move on the objective of this experiment, we decided to test the hypothesis that there is no effect of growing region on the standardized ileal digestibility of amino acids in sunflower meal, and that the standardized ileal digestibility of amino acids in sunflower meal are not different from values that we obtain in sunflower expeller. So now let's move on to the materials and methods that we used to conduct this experiment. We had an 8x11 square design with 8 periods and 8 diets, and as an experimental unit, we had 8 cannulated barrels with an initial body weight of 28 kilo approximately, and for the sample collection, we have a 7-day period with 5 days of adaptation and 2 days of ileal digesta collection. Collecting the samples is very straightforward. We simply take off the cap of the cannula, as you can see in the photo on the bottom right, and then put a plastic bag on the cannula, and then the ileal digesta just flows through. And then we are able to collect that and analyze it for nutrients. So now I would like to quickly explain to you the diets that we used for this experiment. We used the sunflower meal and the sunflower expeller as a whole source of protein in each diet. We had 4 different regions of origin that you can see in the slide, and we received 2 sunflower meals from the United States, 2 from the Ukraine, and 1 from Hungary and 1 from Italy. The sunflower expeller was received from the United States, and it was to complete 7 ingredients for 7 diets. And finally, we had an 8th diet, and it was the end-free diet, and it was made up from cornstarch and sucrose, which means that it was devoid of amino acids. So we use it to calculate the standardized ileal digestibility by determining the basal endogenous losses. We use chromic oxide for all of diets as an indigestible marker. So finally, once we had all of our analysis complete and we had our data, we analyzed it in SAS with the mixed procedure. For this analysis, we used 2 models. For the first one, we had as a main effect the sunflower meal sources and peak and period as a random effect. And for the second one, to compare sunflower expeller with sunflower meal sources, we used a contrast statement. Let's move on to the results, and I want to start by talking about some nutritional characteristics. But before to start, I would like to set up my slides. As you can see, for sunflower meal, we have in light and dark blue sources from the United States. In yellow and orange, we have sources from Ukraine, and in light and dark gray, we have sources from Hungary and Italy, respectively. And finally, in the right side of the slide, we can find the sunflower expeller in light green. So to begin with, I would like to look at acid hydrolyzed ether extract, and we observe big difference in terms of fat content among sunflower meal sources. It could be attributed to the addition of gums during the oil extraction process to the meal by the end of the process, resulting in the increase of oil concentration. Likewise, we can see a big difference in the concentration of fat in the sunflower expeller compared with sunflower meal sources, and it is due to the absence of the solvent extraction step in the oil extraction process to obtain it. And for gross energy, we did not observe big difference in sunflower meal sources, but for sunflower expeller, we observe a greater concentration of energy, and it is because there is a positive correlation between energy and acid hydrolyzed ether extract. So if we look at total dietary fiber, we observe some variations among sunflower meal sources, and it could be possible because of the dehulling process that is dependent on the process of each refining plant. However, we can observe that sunflower expeller has greater concentration of total dietary fiber as well, even more than some of the sunflower meal sources. In addition, this is reflected in the concentration of crude protein. We can see the negative correlation between fiber and protein in this slide. And some of the sunflower meal sources with high concentration of fiber have lower concentration of protein, including the sunflower expeller. And it is because higher concentration of fiber in the diet may release lower concentrations in crude protein. So moving to the results of the digestibility of amino acids. When we look at the 4 limiting amino acids, we did not observe any difference in the standardized ileal digestibility of lysine among sunflower meal sources, but we observed a significant difference in the digestibility of lysine for sunflower expeller compared with sunflower meal. Similar results for the digestibility of methionine. we did not have any difference among sunflower meal, but we had a significant greater digestibility of methionine in sunflower expeller compared with sunflower meal sources. However, for threonine, we observed that the source from Hungary had a significant lower digestibility compared with the sources from US, Italy, and for one of the sources from Ukraine. And for sunflower expeller, we observed a significant greater digestibility of threonine compared with sunflower meals. And finally, for tryptophan, we did not find any significant difference between sunflower expeller and sunflower meal sources. And even we didn't find any significant difference among sunflower meal sources. Finally, to conclude, in terms of nutritional characteristics, the concentration of crude protein and fiber varies among sunflower co-products, and it could be mainly due to the difference in process. In terms of digestibility, only a minor impact of region was observed in the standardized ileal digestibility of amino acids. And in addition, we observed that the standardized ileal digestibility of amino acids in sunflower expeller was greater than in sunflower meal sources. And it could be because of the concentration of fat in the expeller, which reduced the rate of passage in the intestinal tract, enhancing the absorption of nutrients. So with that, I would like to acknowledge Cargill for the funding and the support. And of course, everybody in the Stein Monogastric Nutrition group for the help in this project. And to you, thank you for your attention. And if you have any questions or do you like another information about the work that we are doing in our group, feel free to reach out and visit our website at nutrition.nsi.illinois.edu. Thank you.